This is my first lecture. I am going to start with the general physiology section. In general physiology section, some of the parts will be taken by professors and some parts I will take. So in today's lectures, I am going to start with the topic that is about cytoskeleton, molecular motors and microtubules. So most of us already gone through about the different type of cell organelles in our 12th standard, and though we know about the details of structural and functional details of different type of cell organelles, but still we have little knowledge about the cytoskeleton structures, what are the different types of molecular motors, and what is the structural and functional features of microtubule. So in these lectures, I will try to focus mainly into what are the different types of motor protein and how these motor protein are involved with the different type of cellular activities, and how the microtubule a molecule interact with the motor protein for cellular activities. So let me start with a quote that is about the discovery. For a long time, scientists thought that the inside of a cell was just a jelly-like fluid, no structure at all. But with better microscope, they discovered something incredible, an internal skeleton that gives the cell shape, strength and motion. And that internal structures are nothing, that internal skeleton is the cytoskeleton. So here in these lectures, I will try to explore what are the exact structural basis of the cytoskeleton and what are the motor proteins and how this motor protein interacts with the microtubule. Before proceed to that, let me look into the brief history about the discovery of the cytoskeleton. Before 1960, most of us not aware about the features of the cytoplasms of a eukaryotic cell. So cytoplasm believed to be just a jelly, no internal framework, none. After that, a group of researchers started their research to explore about the natures of cell cytoplasm. So here's Ledwaters and Porters used electron microscopy to spot microtubules in plant cells for the first time in the year of 1963. Right? After that, in 1969 to 1970, another group of researchers started their study to explore more about the features of the cell cytoplasm. So they first time revealed the actin filaments in non-muscle cell. In 1975, there is a discovery of intermediate filaments. In 1980s, it comes that cytoskeletons behaves like a living cell or living part of the cell. In 1990s or till today, what we know that is about the different types of proteins that are molecular protein or motor proteins. The most of the motor proteins that already discovered are the kinesin protein or dynein proteins, and along with myosin proteins also there. So here in these lectures, I will try to expose how these different type of motor proteins involved with the cellular activities. See here, at the very beginnings, what is the basic definition of cytoskeleton? The cytoskeleton actually is a network of fiber that extending throughout the cell cytoplasm. And the most important thing is the cytoskeleton is not a fixed structure. It is very dynamic. And due to that dynamic nature of the cytoskeleton, cell easily can survive in different conditions using the dynamic property of that cytoskeleton structure. So this dynamic nature, it means it can assemble, disassemble and rearrange depending on the needs of the cell. If you see the general functional features of the cytoskeletons mainly provide the mechanical strength to the cell. It also involves to maintain the shape of the cell. It involves the different type of cellular motility like flagellar movement, ciliary movement, amoeboid movement, cytoplasmic streaming. These are different type of a movement that you can see in cell cytoplasms. So these different type of movements involve cell cytoskeleton. Not only this is again another important point that inside cell cytoplasm, the cytoplasmic cell organelles moves from its original positions to the new place based on the necessity. It means these cytoskeleton structures also involves the intracellular transport. So these are the basic functional features that are carried out by cytoskeleton structure. So these are electron microscopic structures of cell cytoskeleton. Now, if you look into the detailed structural features of the cytoskeleton, it is basically composed of different types of fiber, right? So what are the different type of structural element of the cytoskeleton? These are microtubules, microfilaments and intermediate filaments. These three are the backbone of the cytoskeleton structure. They are not only involved with the organizations of the cytoskeleton structures, they are equally important for the function of cytoskeleton. So you can see the structural features of the microtubule and filamentous actin and intermediate filament. If you see the properties for all these three, microtubules specifically determines the positions of the membrane enclosed organelles. It means in cell cytoplasms, organelles needs to fix a specific position to perform its function, and that position of particular cell organelles are maintained by microtubule structures. Along with this, microtubules also involved with the intracellular transport. Microfilaments determines the shape of the cell and which is necessary for the whole cell locomotion or cell motility. On the other hand, intermediate filaments provide mechanical strength and resistance to shear stress. It means all the different components of the cytoskeleton structures are equally important in functional point of view. See here, these are the general characteristics of all those three molecules present in cytoskeleton structures. I'll go into the details of each and every different section. Mainly, I'll focus into microtubule structures today, along with the different type of motor protein. The most important structural features of that microtubule is it is made up of some types of a protein molecule. So what is that protein molecule present in the microtubule structure? These are called the tubulin protein. In microtubule structure, there are two different types of tubulin protein, and they form a dimer, heterodimers, or during the formations of the microtubule structure. These are alpha tubulin and beta tubulin, right? Here, the microtubule structures are basically a cylinder-shaped structure. It contains a lumen, right? You can see here This is alpha-tubulin and beta-tubulin. They form a dimer first, and during the formations of the protofilaments, this tubulin molecule attach with one another and they form a protofilament, right? So here, if you see the diameters of this microtubule molecule, it is around 25 in nanometer, and at the same time, the special features of these microtubules is it can assemble and disassemble. It can polymerize and it also can depolymerize based on the necessity of the cell, right? They are basically found in both the plant cell as well as eukaryotic cell. Now, if you see this structure, it is a cylinder-like structures, and in these cylinder structures, there are numbers of protofilament presence, and those protofilaments are made up of both the alpha and beta tubulin molecule. Another important features of this microtubule is microtubules are polar molecule. Means due to the polarity, it shows two different ends. One is the plus end, one is minus end. So what is the meaning or what is the features of both the plus end and minus end of microtubule molecule? See here. This is the two-dimensional structures of a tubulin protein, alpha and beta tubulin. Initially they form the dimer, and during the formations of these microtubules, they first form a protofilaments by joining one another. Now you can see here, as I mentioned, in the tubulin molecules or in the microtubule molecules, there is two different ends, the plus end and the minus end. The plus end of the microtubules is basically the growing end, while the tubulin molecule binds very rapidly in comparison to minus end, number one. Number two, the plus end actually indicates the growth of the microtubules towards the periphery of the cell, whereas the minus end of the microtubules indicates the origin of the microtubules, like microtubule organizing center that is located in the centrosome. So here the plus end is the fast growing end, where tubulin subunits are added more rapidly, and the minus end is the slow growing end, usually attached to microtubules organizing center or MTOC. So here, during the polymerizations of this microtubule, both the alpha and beta tubulin binds in the plus end, mainly in the periphery. And if you see the depolymerizations or disassembly, it is basically occurs into the minus end. See here, as I mentioned, the microtubules are the structural component of a cytoskeleton, and it not only is structurally important, it also important functional point of view. Here you can see cell motility is the most important functional features where cytoskeletons are actively involved. So here, let me define what is cell motility and what are the different types of cellular movement or cell motility you can see in the cell cytoplasm or in cell. Cell motility is the ability of a cell to move or change its position by organizing its cytoskeleton or using specialized structures like cilia and flagella. In this particular process, there is an interaction of different things. So what are the things? There is an interaction of cytoskeleton with the proteins. It means in the cell motility process, there is an interaction occurs between cytoskeleton and molecular motor protein. And what are those proteins? Those proteins are myosin, kinesin and dynein. And all these three are known as the molecular motor protein. And these motor proteins or the action or functions of these motor proteins is ATP dependent. It means the cell motility where cytoskeleton interact with the molecular motor protein needs energy, and that energy is provided by ATP. In cilia and flagella, the most important type of molecular motor protein involved is the dynein, right? During this motility or flagellar or ciliary movement, what we can see, there is a slide or sliding of the adjacent microtubules against each other that produce a wave-like motions that moves the cell or fluid. This is the most important features, how cilia or flagella or moves by using certain types of molecular motor protein like dynein here. So here there is a sliding of microtubules against each other in presence of this dynein motor protein. The same things you can see in the muscle during muscle contraction. There is a sliding of filament, actin and myosin filament slide over each other, leads to the contractions of the muscle. And during that sliding, the protein involved myosin protein. You can see here, this is the one microtubules, this is another one, and these are the motor protein. And these motor proteins or actions of those motor proteins are ATP dependent, that is powered by ATP. And during the cellular movements, there is a sliding occurs between the adjacent microtubules that allows the cellular movements, as well as it also allows the movements of or move past the fluid of cell. Now, see here, as I mentioned, that inside cell cytoplasm, there are a number of cell organelles. And based on the necessity of the cell, they can move from one place to the new place. But for the movements or for the transport of those cell organelles, they need a track. They need a vehicle, right? So here cell organelles act like a cargo and motor proteins acts like a vehicle and microtubules acts like a path or track. So all these three components involved with the working of cell organelles inside cell cytoplasm. The organelles are transported along microtubules or microfilaments by motor molecules that is attached to their receptors. You can see here, this is the microtubule structures again. The surface of these microtubule structures, this is the molecular motor protein. And one part of this molecular motor proteins is attached to the microtubule structure, and another part which contains the receptors is attached to the cargo, that is cell organelles here. So once this cargo binds with these motor proteins, these motor proteins are based on the type of motor proteins. These motor proteins either work towards the plus end of the microtubules or the minus end, right? Because a different type of molecular motor protein shows different type of transport. Like we can see anterograde transport and retrograde transport. In case of anterograde transport, the most important type of motor protein involved is kinesin. In this particular case or in anterograde transport, what we see, this kinesin protein carries the cargo towards the periphery of the cell from the center, or it carries the cargo towards the plasma and to the microtubule or along the microtubule structure. So it means based on the type of protein attached here, we can see two different types of transport mainly. One is called anterograde transport, where the cargo moves towards the plasma of the microtubule, and another is the retrograde transport, where the cargo moves toward the minus end of the microtubules by motor proteins along the microtubule structure. So you can see here common example that we can see how the organelles works. Migrations of neurotransmitters containing vesicles to the tips of nerve cell axon. Yes, if you see the structures of the neuron, the neurotransmitter molecules are basically present in the synaptic vesicle, and that neurotransmitters are transported from the cell body towards the axon terminal. And that transport of neurotransmitter or the transport of vesicles containing neurotransmitters actually carried out by the microtubule molecule. And here you can see these are the motor protein which is attached to the vesicle, and they move towards the axon terminal from cell body. Here the cell body actually indicates it is the center part of the cell, and axonal terminals is the peripheral area. So it means this is the anterograde transport, where the synaptic vesicles moves from cell body towards the axon terminal. And another way you can see, there is a free vesicle that moves towards the opposite ends. If it moves towards the opposite ends, it is nothing, it is the retrograde. So both the anterograde and retrograde transpose, you can see both anterograde and retrograde transpose is used to explain how the different type of cell organelles, how the different types of vesicular structures can move inside cell. See here, the microtubule, as I mentioned, it is a very dynamic structure. It can assemble or polymerize, it can disassemble or disassemble or depolymerize. Both the polymerization and depolymerizations depend on the requirements of the cell. So during the polymerizations, what is the most important features here? There is a binding of the tubulin dimer in one end, and that binding in one end, there is a binding, in another end there is a detachment of that tubulin molecules. Both the binding and detachments of that tubular molecules depends on phosphorylation, dephosphorylation events. It means for the polymerizations, there is an active involvement of certain types of energy molecules, mainly the GTP. It grows at each end by the polymerizations of tubulin dimers, that is powered by GTP. In another ends, there is a shrink at each end by the release of tubulin dimers. So here's both the events of polymerizations and depolymerizations are very, very important. That is specifically you can see in the cellular motility or cell motility. It participates in a wide variety of cell activities, mostly involved in the motion or motility, and the motions is provided by a protein, that is the motor proteins, that is powered by ATP. You can see from here also the polymerization process as well as the depolymerization process. This is the post state of microtubules, which is a cylinder shape, and In the growing microtubules, there is a bindings of that dimer, and during that binding process, there is active involvement of energy molecule, which is provided by ZTP. Now, what is the specific features of all those different type of motor protein? As I mentioned, the most important type of motor proteins are kinesin, dynein, along with the myosin. So here you can see two important, one, the kinesin and the dynein. The kinesin protein is the most of this moves towards the plus and the microtubules. It means it is involved with the anterograde transport process, whereas the dynein proteins, which moves towards the minus n, means it involves with the retrograde transport. The common examples, as I've already mentioned, the rapid transport of organelles like vesicles and mitochondria along the axons of the neuron takes place along the microtubules with their plus n pointed towards the end of the axon, and here the motor proteins is kinesin. And the same things you can see during cell division. Both the mitotic and meiotic cell divisions, what you can see in a particular phase of the mitotic, in a particular phase of cell divisions, there is a movement of chromosomes towards the pole, and that chromosomal movement towards the pole is basically carried out by certain types of protein. So here the migrations of the chromosome in mitosis and meiosis takes place on microtubules that make up the spindle fibers. Here the most important type of proteins involved is dynein. So it means in cell cytoplasm or in cell, there are numbers of activities is going on, and all those activities involves different types of protein with their specific function. See here how actually it happens, or how actually the cargo moves towards the pole. Once this binds with this tubulin dimer, or it binds with the microtubules, based on the natures of that protein, it moves. The same things you can see here, two different types of proteins. One is the kinesin, another is the dynein, and there is a organelles that binds to this protein, and they move towards the plus ends, means they show the anterograde one. At the same time, another protein, that is the dynein, it shows the retrograde one. The same things you can see here. During the segregations of the chromosome, the spindle fibers form, and this spindle fiber allows the migration of these chromosomes towards the pole. So in general, now we can summarize what is the main functional features of the microtubule. It gives proper shape and support to the cell. It resists the compression. It serves as a track along which organelles can move via the motor molecule. It is responsible for the separation of chromosome during cell division. It is an active component of centrosome, centriole, and Cilia flagella. So these are the basic functional features of the microtubule. This is how different types of molecular protein involved with the transport process of different cell organelles inside cell cytoplasm. Now let me move with the basic understandings of the centrosome, centrioles, cilia, and flagella. What is the basic definitions, or what is the role of centrosome in cell? Centrosomes is a non-membranous organelles, which is basically known as microtubule organizing centers, from where microtubule originate, right? It's the region near the nucleus from which microtubule starts growing. And that centrosome, you can see here, that centrosomes is basically made of a pair of centrioles. Now we'll see what is the exact structural basis of the centriole. Here, see, this a centriole are mainly made up of the microtubule fiber. In a single centriole structures, you can see it is composed of 9 sets of triplet microtubules arranged in a ring. See here, this is a single triplet. This is another single triplet microtubule. Like this, in a centriole structure or a single centriole structure contains 9 sets of triplet microtubule, and they make a ring-shaped structure. So here centrosome are mainly composed of a pairs of centriole, and that centriole are made up of microtubules, mainly 9 sets of triplet microtubules arranged in a ring. And that centrioles is very vital because it replicates before cell divisions, which is essentials for the divisions or migrations of cell towards the opposite side of the two different cells during the divisions. It also involves the organizations of the microtubule assembly, but it is not the main function. So what is the most important characteristics of centrioles, and how these centrioles equally important with the locomotion process? Now we'll see. As I mentioned, that centriole contains the microtubule domain. Like this, if you see the structures of cilia and flagella, they also contains the microtubule domain. And due to the presence of those microtubule domains in the cilia and flagella structures, they allows the cilia and flagella to move like a wave-like motion, right? See here, both the cilia and flagella are constructed from microtubules. Both provides either locomotions for the cell or move fluid past the cell. Cilia and flagella differs in their beating pattern. Yes, if you look into the motion of both the cilia and flagella, their beating patterns are different. One moves like a wave, right? And in case of flagellar movements, the force that is generated during the motions, it is along the axis of the flagella, right? But in case of cilia, cilia sweep mucus carrying trapped debris from the lungs by a different type of motion. So like a movement of a boat using, it's like an oar. The motions of the cilia is like a oar, right? You can see a flagellum has a wave-like motions that generates force in the same directions as the flagellum axis. That is very common that you can see in the movements of the sperm cell, and the directions of the movements here is unidirectional, and during the movements of or during the motions of the sperm cells, it generates force in the towards the directions it moves. On the other hand, if you see the ciliary movements, you can see two different type of things here. This one is called an active stroke, and another one is a recovery stroke. This is like a works of force. So here, in both the ciliary movements and flagella movements, involves the microtubule structures, and both the ciliary movements and flagella movements involves with the molecular motor protein. So here motor protein again acts as a vehicle. The microtubule acts as a track, and like what we have seen in the ciliary movements, it clears the debris from the lung tissue or respiratory passage. So here, that debris here is nothing like a caribou. Now see, what is the exact arrangements of the microtubules in ciliary structures. In ciliary structures, you can see there are a number of microtubule or doublet micro. These are called doublet microtubule. That is just around the outside of the doublet micro. There is a cell cytoplasm. Means these doublet microtubules are present in the periphery, which is just we can say it is connected with the cell cytoplasm. At the center of these ciliary structures, there is a pair of microtubule. So if you look into the arrangements of the microtubule in the cilia, it is called 9+2 arrangement. So what is the meaning of this 9+2 arrangements of the microtubule that is found in the ciliary structures? At the periphery, there is a 9 pairs of doublet microtubule arranged. At the center, there is a pair of microtubules. And this doublet microtubule or adjacent doublet microtubules attached to one another by specific type of motor protein. These are called the Dynein motor protein, right? Other than this, there is a connections between the central microtubule, and that central connections is called central bridge. So this is what is the general structural organizations of the microtubules present in the ciliary molecule or ciliary structures. Here's another important things. This is called the basal body, and the presence of that basal body in the ciliary structures. This basal body has the same structures as the centrioles, and that basal body actually helps in the attachments of both the cilia and flagella to the cell. So basal body also has some structural importance because it actually allows the attachments of both the cilia and flagella to the cell. You can see here the motor molecules that is present here between the microtubule molecules is the dynein that helps in the attachments, and that dynein is responsible for the bindings of both the cilia and flagella during ciliary or flagellar movement. And this ciliary and flagellar movements also need some energy. And if you see the mechanism involved or the whole process of the ciliary and flagellar movements, how the ATP molecules are involved, here's the binding of adjacent microtubules by dynein proteins actually allows the binding of microtubule during the motions, and the binding is basically depends on the sufficient supply of energy that is provided by ATP, and this ATP powered activity or ATP powered alterations of structural change of microtubules actually allows the motion or movements of cilia molecules. So in this process, whole process, you can see the structural changes. Due to the structural changes, this molecular motor protein that is attached in between the microtubules grabs the nearby microtubule. So as I mentioned that in the ciliary structures, the microtubules are arranged in a specific pattern, and adjacent microtubules attached to one another by the dynein protein. And here, during movements, how the dynein played an important role. During this process, when ATP supplies, there is a conformational changes occurs into the motor protein, and due to the conformational changes in the motor protein, it allows the motor protein to grab the adjacent microtubule, right? So it first grabs the nearby microtubule, and after that it pulls or slides it, and then it releases it. So this process happens again and again during the ciliary or flagellar movements. So here's the most important thing is the conformational changes of the Dynein protein. Once the conformational changes occurs, then only by the process of assembly and disassembly of the microtubules, it allows the movements of cilia. Now, another important part, that is the structural component of cytoskeleton, is the microfilament. So I'm just giving this a brief structural demonstrations about the microfilament. Like microtubules, microfilaments is also important, and if you see the microfilament structures, it has a specific diameters. It is around 7 nanometer, and it is basically made up of different types of protein. Mainly 2 different types of proteins are present in microfilament structures. One is called Z protein or globular protein, and another one is F proteins or F actin. Actually, this is filamentous actin. So here's like microtubule structures, microfilaments also having 2 different end. One is plus end, you can see, and another is the minus end. So in the plus end, the polymerization occurs by the attachments of the globular actin in presence of the energy molecule, and the depolymerization occurs in the minus ends or the microfilament structures. So during the polymerization process, what we see in a single microfilament structures, it is actually like a chain, and it is a twisted chain, and in this twisted chain numbers of globular proteins attached. Once they attached into the twisted chains, they become or they transform into filamentous from globular actin. So here's this microfilament is very vital in different type of cellular activities, like we can see the involvement of myosin in the muscle contraction, right? So not only microtubules, along with this microfilaments also equally important in functional point of view. Here you can see the microfilaments forms tight bundles that support the core of microvilli, and this microvilli is nothing, it is a finger-like projections on the cell surface. These structures increase the cell surface area, that is very important for the particular cell, mainly the cells present in the intestines and kidney, because due to the increase in the surface area, it helps in the absorption and transport of different materials efficiently. So here, due to the presence of those microfilaments bundled into the cell surface, it increases the cell surface area that ultimately allows some physiological process like cellular absorption as well as the transport of different materials across the cell surface. Another important property of cytoskeleton is cytoplasmic streaming. So what is cytoplasm streaming? It's also like a motion, but it is very specific. It is very specific. Here you can see the molecules, how the molecules, different type of molecules moves inside the cell cytoplasm. It is a circular flow of cytoplasm within the cell. This movement is a result of actin-myosin interactions, and it is mainly due to the transformations of 2 different states called sol and gel state, and this sol and gel state transformations actually allows the cytoplasmic streamings or the flow of different molecules across the cell cytoplasms. Not only this, due to this cytoplasmic streaming, it actually helps to maintain the concentrations of different type of molecules across the cell cytoplasms, which is essential for proper cell functions or cellular functions. So up to this, what we have seen, there are a number of structures that played an important role in different points. So those structures are the microtubule structures, microfilament structures, and another important is the intermediate filaments. I have not discussed yet. There are numbers of proteins involved in this whole process, mainly the kinesin protein and the dynein protein. The functions of those proteins basically ATP dependent. At the same time, we have seen the whole process or the movements of different type of cell organelles inside cell not only depends on protein, obviously certain other factors also equally influence. The factors is the necessity of the cell. Along with this, we can say the proteins or molecular motor proteins are clinically also very important, because if I take one example to explain this, what we see in the nerve cell degeneration, there is a loss of the nerve cell, no transmissions of transmitter. So as we have already seen, the nerve cell transmissions involves a certain types of protein, but the degenerations of the nerve cells means dysfunctions of those proteins that ultimately affects the whole transmission process, that promotes that influence the degenerations more rapidly. So this is just a silly example how to explain about the importance of the motor proteins in clinical aspect. So this is all about in today's lecture.